Hello everyone, this is Dr. Nasser and I would like to tell you today about a unique red blood cell morphology known as spherocytes. In this image, you do see some cells that are smaller in size, they're round, their color is slightly darker than other cells that are around. There is no central pallor present. These cells are referred to as spherocytes. A patient may have spherocytes in their blood smear for various reasons. Some of these reasons may be hereditary because of the mutation in the gene that codes for the protein that are associated with red blood cell membrane. When there is a problem with one of those proteins such as the spectrins, alpha and beta spectrin, protein 4.2, anchoring, these group of proteins stabilize the red blood cell membrane. And when you have a genetic mutation, the protein product of that mutation, if there's any, is unable to perform the same task. Therefore, the membrane is going to be weak and there is going to be constant loss of membrane without losing the volume. Therefore, the cell is going to become spherical, dense red because of a higher concentration of hemoglobin, a shorter lifespan than normal cells. Because of the loss of the surface area, the red blood cell loses its deformability and is unable to navigate into small blood vessels such as small arterioles and capillaries. And usually is removed by the spleen. Therefore, their lifespan is going to be much shorter. Sometimes the spherocytes are present on blood smear, like the case for the smear that we are looking at, due to an autoimmune hemolytic anemia that is due to IgG antibodies. This type of anemia causes the red blood cells to be removed in the spleen because the red blood cells are coated by antibodies in blood, and when that blood circulates through the hemodynamic of spleen, macrophages, splenic macrophages will try to remove the antibodies, and some of the membrane of the cell that is bound to the antibody will be removed. Therefore, the cell starts losing its surface area without losing much volume, and therefore acquires this morphology of spherocytes. Also, you can see spherocytes in severe cases of burn. And in those cases of burn, you will have a lot of schistocytes in the smear as well. Schistocytes and spherocytes are commonly seen within 24 hours to 48 hours after severe burn, and they will be removed as long as the spleen is intact and healthy. They will be removed very quickly from the circulation, so it is often seen immediately after the incident. In here, you do see the anisocytosis is present. I do see some polychromasia in here, as well as a couple other abnormal morphology, which we will allocate a different video clip for each one of these morphologies and discuss it in greater detail. There is also a couple Hal Jolly bodies that I do see present here, and perhaps the third one might be in here. You can see the platelets. There are a lot of artifacts on this slide. These are all artifacts in this area. Platelet, 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 artifact. And this is the characteristic of spherocytes. Small, round, dense, red blood cell with no central power. And in this case, when we are looking at this case is a result of autoimmune hemolytic anemia. And polychromasia indicates the attempt by the bone marrow to compensate for low RBC count in the blood.